Okay, today we're going to talk about microbial growth and specifically bacterial growth. And this will be the last lecture that will be on the exam, which is Wednesday. Monday's class is not on the exam. So when it comes to microbial growth and specifically bacterial growth, we're talking about the increase in the number of cells, not the size of a cell. We don't start out with little baby bacteria, and then they go through the terrible twos, and then they're juveniles, and then finally adults. When the bacteria divide in two, you have two complete cells. So growth is an increase in the number of cells, not the size of cells. There are many factors that affect microbial growth, and the same kinds of factors that affect all kinds of living things like temperature. Every microorganism has a minimum temperature at which they can still grow, an optimal temperature where they grow the best, and a maximum temperature they can tolerate and still grow. And this does vary from organism to organism, but we can classify microorganisms, and in particular bacteria, based on temperature requirements. So the mesophiles, meso means middle, so this is the mid-range of temperatures, around 37 degrees Celsius. And what did we say was the significance of 37 degrees Celsius?
My body temperature.
Right, so it's 98.6 degrees Fahrenheit. And so many microorganisms like it around 37 degrees Celsius. They can tolerate warmer, maybe up to 45 degrees Celsius. And of course, they can tolerate cooler. They'll grow more slowly, but they will still be able to grow. The psychrotrophs, they like room temperature, like around 23 degrees Celsius, 73 to 75 degrees Fahrenheit. That's the ideal temperature for them, but our body temperature is way too hot for them, so they would die. And they will grow in the cold, including in the refrigerator. And psychrotrophs are responsible for spoiling foods. Psychrophiles are true cold-loving microorganisms, and they like to grow in the cold. In fact, they need to grow in the cold. Room temperature will kill a psychrophile. It's just too hot for them. So if you want to be a researcher and work with psychrophiles, you better love the cold, because you have to work in a giant refrigerator in order to work with these microorganisms, because they must be kept cold. On the other end, there are some bacteria that like it hot, and these are thermophiles, and usually right around 62, 63 degrees Celsius. And if we were meeting for lab, I would be able to demonstrate this, because the hot plate that we use to dry our bacterial smears is kept around 60 degrees Celsius. And so you can actually put your hand on it for 2 or 3 seconds, but then you have to pull away, otherwise your hand will burn. So it's hot enough, to burn us or to scald us if we were in that temperature water. But that's what some bacteria can grow in, right? The thermophiles. So they do like it very, very warm. And that's sort of the upper limit for true bacteria. If you want to get to extremely high temperatures like boiling, then you have the hyperthermophiles. And these are typically members of the archaea. So not true bacteria, but the archaea. And they can grow in boiling water. That's their optimum temperature. So life can exist at extremely high temperatures. Usually not multicellular life, but single-cell life forms can. And when it comes to all these different temperature requirements, what is it specifically about the cells? What is in the cells that actually determines the temperature requirement? So what is it about the cells that actually determines their temperature requirement?
Is it their genetics?
Now there's things inside the cells What things inside the cells actually determine the temperature requirement? Enzymes, right, the enzymes and proteins. And we see from hyperthermophiles that enzymes can be extraordinarily stable, right? Because most enzymes denature with heat, but with hyperthermophiles, their enzymes are extremely stable. Do you want your enzymes to be that stable? I mean, we can clearly have enzymes exist that are remarkably stable, but is that something that cells want? I mean, do they want their enzymes to last forever? Would that be a good thing? No, not always. In fact, rarely do they want that to happen because making enzymes that do eventually degrade is important. It helps the cells to regulate metabolic pathways because they have to constantly synthesize new enzymes. And that's the way to help regulate, right, those pathways. If the enzymes never got denatured or destroyed, they would keep working forever, and a cell might not want that pathway to be active. So we know we can make enzymes very stable, but most cells, their enzymes are not very stable. And they want that turnover rate so that they can regulate pathways. So when it comes to the actual temperatures, these ranges are approximate. It varies slightly from organism to organism, but psychrophiles do like it cold, and they can grow at freezing. And remember, water can be liquid at 0 degrees if it has enough solutes in it, like salt. So as long as water is liquid, life can sometimes grow below the freezing temperature of 0 degrees Celsius. Psychotrophs like it around room temperature, up to maybe 30 degrees, but they tolerate growing in the cold. And because they tolerate the cold, they can spoil food in your refrigerator. So if you give your food long enough, it will still go bad. Microorganisms will still grow in the refrigerator, just more slowly. Mesophiles like it around 37 degrees, so most of our pathogens are mesophiles because they like it around 37, and thermophiles like it hot, right around 60, 62 degrees Celsius. But you can't go too much beyond that and still be true bacteria. You have to belong to the archaea to get above 70 degrees Celsius. Now, psychotrophs, again, they do like room temperature, they tolerate growing in the cold, and they do spoil food. Now, it's getting a bit chilly, faster than expected outside, so most people are not going to be having picnics anymore, but the problem with picnics was that food is sitting out in the warm summer day in the sun, and so if your hot food cools down or your cold food warms up, then microorganisms will start to grow. And so you do have to be concerned about what's called the danger zone, because in this temperature range, many microorganisms grow very quickly, and they often produce toxins, which is what causes food poisoning. So the main idea is to keep hot foods hot as long as possible and cold foods cold as long as possible before you eat them. Because if they warm up or cool down, microorganisms will start to grow, and that increases the risk of food poisoning. pH is another very important physical factor. Most organisms like it around pH 7. And what is pH 7?
Neutral.
Around neutral. So most living things do like it around neutral, but there are some organisms that like it more acidic, like fungi, molds, and yeasts, right? They like it between pH 5 and 6. But there are things that do like it very acidic. And the only thing that comes to mind is a vinegar eel. And a vinegar eel is a very tiny worm. It's about 1 to 2 micrometers long. So you can see them with your eyes if your eyes are still good. But they grow in vinegar. And vinegar is very acidic, but that's where those vinegar eels grow. So there are organisms that are adapted to a more acidic environment, as well as others more adapted to a more basic environment. But most living things do like it around a pH 7. And again, that also has to do with their enzymes. Osmotic pressure is important. When you increase the solute concentration, you create a hypertonic environment. You can do that with salts or sugars, and that sucks the water out of cells. And for bacterial cells, that means the cell membrane pulls away from the cell wall, and we call that plasmolysis. May or may not kill the bacteria. It might just put them into suspended animation. Now, there are organisms that like salt. These are extreme or obligate halophiles, so they love salt. They actually need that high osmotic pressure. That's what they evolved with. There are other microorganisms that tolerate the higher salt concentrations, and these are called facultative halophiles. So when you see the word facultative, think tolerates. And so a facultative halophile can tolerate a higher osmotic pressure, at least for a while. So what are some typical salt concentrations? Well, in our body, in our tissue fluids, the salt concentration is technically 0.85%, so 0.9% just to round it up. So it's less than 1% salt. The ocean is 3%. If you've ever been to the ocean and tasted seawater, you know it tastes really, really salty. That's only 3%. But there are bacteria, well, when it comes to the ocean, you know, some people think because it is salty that makes the ocean cleaner, right? They don't have to worry about microorganisms, when in fact the ocean has countless millions and billions of bacteria and viruses per milliliter of seawater. In fact, the ocean is one of the richest environments for microorganisms on the earth. Of course, almost none of those can hurt you, so you don't have to worry about them. But sometimes they do close beaches because of sewage contamination, because fecal bacteria can tolerate the ocean salt, at least for a while. So there are some pathogens which are facultative halophiles, so sometimes they do have to close beaches because of sewage contamination. So the ocean is not what you call clean or cleansing, because it's full of microorganisms. And we do have some areas on the earth with extremely high salt concentrations, like the Dead Sea and the Great Salt Lake. And the concentration of salt can be anywhere from 15%, even up to the saturation point, around 30%. And we find microbial life here. There are obligate halophiles that live in these bodies of water. Now, where do we find the Dead Sea in general? Where do we find that? Where's that located? What region of the world? I know you've heard of the Dead Sea. In the Middle East, right, Rachel? So in the Middle East. And what is it that you cannot do in the Dead Sea? What cannot happen in the Dead Sea? Oh no, you can swim. Lots of people go to the Dead Sea to swim. But what can you not do when you're in the water? Oh, sure, a cut's fine. It's going to sting a little bit, but it'll be fine. Right, you cannot sink because the concentration of salt is so high that the water is more dense than you are. So you float in the Dead Sea, so you can't sink, right, because it's so salty. I had a student one time, he was Israeli, and he went back to Israel for spring break. And he went to the Dead Sea and he brought back a sample of water. And I made up some salty agar plates with 15% salt, and we were able to isolate 3 different halophiles from that Dead Sea water. So there is microbial life there. Would you have to worry about any of those microbes being pathogens and causing disease in you?
No, because they wouldn't be able to survive.
Right, they couldn't survive in you because you're so dilute, you're so hypotonic that there's no way a Dead Sea microorganism is going to be able to live in you. And then we have the Great Salt Lake. Where's the Great Salt Lake? Yeah, where in the USA? It starts with U. Utah, right? So Utah. And again, these bodies of water do contain microorganisms. So life can exist almost anywhere as long as there's liquid water. So just to remind you, here we have a bacterial cell in an isotonic environment, so it's happy. In a hypertonic environment, like 10% salt, the membrane shrinks, it pulls away from the cell wall. We call that plasmolysis. May or may not kill the bacteria. It might just put them into suspended animation, and they come back to life when conditions become favorable. Of course, we're all carbon-based life forms. As far as we know, all life is based on carbon, right? Carbon forms the backbone for all the macromolecules, the hydrocarbons. Chemoheterotrophs use organic carbon sources for energy and for carbon. Autotrophs, remember auto thinks CO2, so they use carbon dioxide. What are NS and P?
Is it nitrogen, sulfur, and phosphorus?
Yep, all very important elements. And there are many, many trace elements as well. And the trace elements are inorganic compounds like iron, manganese, zinc, and they're typically, found in the environment without any problem. And they're often enzyme cofactors. Remember to make the enzymes complete and functional. When it comes to oxygen, do all forms of life require oxygen to live?
No, like anaerobes don't.
Right, no, they don't. But when it comes to microorganisms, we can actually test for their oxygen requirements using a fairly simple test with melted agar deeps. And this is something we would have done in lab, and we'll still talk about the procedure in lab when we get to it. But the idea is you take the agar, you boil it, then you cool it down and hold it at about 47 degrees Celsius, so that way it stays melted, but it's not so hot that it will kill the bacteria that we put in there. And then you rotate the tubes between your palms to mix the bacteria with the agar, and then let the tube cool and solidify, and it traps the bacteria in place. A concentration gradient of oxygen forms in the agar deep, and the agar becomes completely anaerobic barely halfway down the agar deep. So not much oxygen can diffuse through the agar. But based on where the bacteria are growing, you can determine the oxygen requirements and classify them. So obligate aerobes must have full atmospheric concentrations of oxygen. These diagrams are not accurate. An obligate aerobe will actually only be growing on the surface of the agar, nothing like what's shown here. It's not down in the agar, just on the surface of the agar. A facultative anaerobe, what did I just say you should think when you see the word facultative? Right, that it tolerates. And so what a facultative anaerobe tolerates is the anaerobic environment. So facultative anaerobes love oxygen and they use it, but they do not need it. So the way they look in the agar deep is you have a lot of growth on the surface, but also growth all throughout the agar, even when where there's no oxygen, because they don't need it. They can grow without it. So facultative anaerobes love oxygen, but they don't need it, right? They can grow anaerobically. An obligate anaerobe, however, must have an anaerobic environment. There cannot be any oxygen present because oxygen actually kills an obligate anaerobe. So they only grow in the bottom half of the agar deep. Aerotolerant anaerobes are different. These are anaerobic microorganisms, so they do not use oxygen, but they can survive in the presence of oxygen. And so an agar deep would show growth all throughout the agar, but nothing especially good on top. So it's not a facultative anaerobe. Facultative anaerobes love oxygen, lots of growth on the surface. Aerotolerant anaerobes, no special growth on the surface, but lots of growth all throughout the agar deep. And lastly, we have microaerophiles. So these organisms need a little bit of oxygen, but too much will overwhelm and kill them. And they actually grow just beneath the surface, so not this far down, but just beneath the surface. So they need some oxygen, but full atmospheric concentrations will kill those organisms. So this is how we can determine the oxygen requirement and classify bacteria. Now, you heard me say for obligate anaerobes and microaerophiles, oxygen can kill them. How can oxygen kill? I mean, oxygen is so important and necessary for so many life forms, and yet oxygen is one of the most toxic substances on the earth. It is absolutely toxic, and yet so many organisms need it. So why is oxygen so toxic? Well, it produces many toxic forms, in particular free radicals. And you may recall that a free radical is a molecule that has unpaired electrons. And remember, electrons do not like to be alone, right? They like to be paired up. So these molecules attack other molecules inside of cells in order to satisfy their lonely electrons. So a free radical can attack nucleic acids, DNA, RNA, they can attack proteins, lipids, all kinds of things, and do damage inside the cell. So oxygen produces a lot of these toxic free radicals. So organisms that use oxygen or simply tolerate its presence need to have a way to deal with these toxic oxygen molecules. And the way that these organisms deal with that is with enzymes. So, and when this converted, this kind of got messed up here, but the hydroxyl radical is down below here. So there are these toxic forms of oxygen. And one of the most toxic is superoxides. And there are different superoxides, which is why this is not a balanced equation, because the actual superoxide varies. But superoxides are very, very toxic. So microorganisms have enzymes like superoxide dismutase to convert those toxic superoxides into something else which is still toxic. What is H2O2? What's H2O2? Hydrogen peroxide. So hydrogen peroxide is not a free radical, but it does cause other free radicals to form. So hydrogen peroxide is still very toxic. So cells need another enzyme to break down hydrogen peroxide. And there are two possible enzymes, catalase and peroxidase. They both break down hydrogen peroxide. Now, one of these enzymes we can test for in lab. Which enzyme and why? Which of those two enzymes, catalase or peroxidase, can we test for in lab, and why can we test for it? No, which of the two enzymes can we test for, Christina? Catalase or peroxidase? Right, so we can test for catalysts, right? So then how do we know?
Because it's found in almost every living thing.
Well, and also peroxidase is found in a lot of living things too, but in lab, how do we know it's catalase and not peroxidase?
Is it because he adds more oxygen?
Right, because we can see bubbles of oxygen gas. And so when you mix peroxide with bacteria that produce catalase, you see bubbles. If the bacteria have peroxidase, they make more water. You can't see more water being made, but we can see bubbles of oxygen gas. And if you've ever put a peroxide on a cut or a scrape, you've seen that it bubbles because our blood and tissues are full of catalase, right? Because we are very aerobic organisms. We need a lot of oxygen. And so we produce a lot of toxic hydrogen peroxide. So our cells and tissues are full of catalase. But the main idea here is oxygen is very toxic. So cells need a way to deal with the toxic free radicals produced by oxygen. And that way is with enzymes. So whether they actually use the oxygen for cell respiration or they're just growing in the presence of oxygen, they still need these kinds of enzymes to deal with the toxic oxygen-free radicals. A variety of organic growth factors are required. Many microorganisms synthesize almost everything they need, but whatever they don't need, they usually have to find in the environment. So things like vitamins, amino acids, purines, and pyrimidines. What are purines and pyrimidines? We don't see those two words very often, but what are they? What are purines and pyrimidines?
Is purines DNA?
Well, they are the nitrogenous bases used to create nucleotides for DNA and RNA, right? So some microorganisms can synthesize almost everything they need. Others have to get them from the environment. When we grow microorganisms, we need a culture medium, singular, media plural, and it's simply nutrients that support microbial growth. Once you have your growth medium, you have to sterilize it because the only things you want to grow in it are the things that you put in there. And the things you put in there is called an inoculum. So a loop full of bacteria is an inoculum. And then you have a culture of microorganisms growing. Now, broth is easy because it's basically soup, right? A liquid medium containing nutrients. But as you recall, we do like to see colonies growing on the agar surface, right? Because we can see characteristics that can help us to tell how many different kinds of microbes are present and potentially what they might be. But the problem was finding a solid medium that could be used to grow bacteria. In the very beginning, they actually tried things like potato slices and grew bacteria on potato slices. But that obviously wasn't very good because lots of bacteria love to eat starch. And then they tried gelatin. The problem with gelatin is it has a very low melting point. It actually melts at 37 degrees, which is what many microbes like to grow at. So that didn't work. Plus, a lot of bacteria eat gelatin. They digest it. So that didn't work either. So finally, they found agar, a complex polysaccharide extracted from seaweed. And it's an unusual polysaccharide. Very few microorganisms can digest it. And you do boil it to make it liquid. So it stays solid at very high temperatures. And so agar, became the standard that was used. You just take your liquid broth, you mix it with some agar, you boil it, and now you have a solid agar medium. It does start to solidify around 40 degrees C. So as I mentioned, when we use melted agar deeps in lab, we try to keep them at around 47 degrees Celsius. That way they stay nice and liquid, but it's not too hot to kill the bacteria that we're going to stick in there. Because once the agar starts to get thick and liquefy, the only way, or solidify, the only way to back into a liquid is to boil it again. There are different types of culture media. We have chemically defined media, also known as synthetic media, and complex media, which is non-synthetic. Almost all media are complex. They are not synthetic. A synthetic medium, you know the exact chemical composition. So there's very few types of media like that because microorganisms need all different kinds of nutrients. So the only way to get them is by using extracts and digests of things like yeast, meat, or plant. So for example, you have brain heart infusion agar, where they actually take cow brains and hearts and grind them up and cook them up and use them to make a nutrient medium, right? We don't know exactly what's in there. There's all kinds of organic compounds and growth factors and all kinds of other things. So we don't know the exact chemical composition. One of my favorite types of agar is chocolate agar. You ever heard of chocolate agar? It's nice and brown like chocolate, but it sure doesn't taste like chocolate because chocolate agar is just cooked blood. When you cook blood, it turns brown. So chocolate agar is a blood agar plate that's been cooked, so it's brown. But lots of nutrients in blood. So here we have an example of a synthetic medium. You measure out each ingredient, you know exactly what's in there, but this cannot support the growth of very many microorganisms. So we use complex media. So peptone is partially digested protein. Beef extract, we don't know exactly what's in there, and that's what makes it complex. There are microorganisms that are anaerobic, especially the obligate anaerobes, which can't tolerate iodine because it will kill them. So you can make special media called reducing media, which use chemicals like thioglycolate or oxyurase, which bind to the oxygen so that it keeps it away from the bacteria. We always heat the medium because hot liquids cannot hold much oxygen. You know, cooler liquids hold a lot more oxygen. You can use special little chambers like anaerobic jars, something we actually would have done in lab. And this is interesting because you put your cultures in there and then you add these chemical packets. And so the oxygen is actually removed chemically. Hydrogen gets produced, and there's a catalyst that combines the hydrogen with oxygen in the jar to make water. And that's how the oxygen gets removed. It's converted into water. So it's pretty interesting. If you do a lot of work with very sensitive anaerobes, then you need a larger chamber. With the glove hands, you put your hands through in the glove, so the whole chamber is anaerobic. And that way, you don't have to worry about your sensitive anaerobes being killed, with exposure to oxygen. Capnophiles are anaerobes that do like a lot of CO2, so they're very autophagic. They need lots of CO2. And in the old days, and we're talking a long, long time ago, they use candle jars. So they actually would light a candle, put it in the jar, seal the jar. As the candle burned, it would consume the oxygen, making it anaerobic. And as the wax burned, it would produce CO2 and enrich the environment. These days, we have chemical packages or special incubators that have a high CO2 content. Now, when you obtain a sample from a patient or from the environment, there's always more than one organism present. So you need a way to separate, right, those different microorganisms. One way to do that is by using selective media. A selective medium contains chemicals that inhibit one group of microorganisms and allows another to grow. So for example, you may have a selective medium that inhibits gram-positive bacteria, but allows gram-negative bacteria to grow. In that way, you can selectively isolate your gram-negative bacteria from that sample. Differential medium may or may not let a wide variety of microorganisms grow, but as they grow, they typically exhibit a characteristic like lactose fermentation. And so you will see a color change in the colony, and that tells you those bacteria are fermenting lactose. So there are many different kinds of differential media. They usually show some kind of color change, which indicates some kind of biochemical activity occurring, like lactose fermentation. It is possible to combine both selective and differential properties in the same agar plate. So a medium can be both selective and differential. And we will talk about selective and differential media in lab in the coming weeks. Any questions so far?
No, but it's really cool how they have all these different ways to...
Yes, and unfortunately, we're just going to look at pictures and talk about them, not actually play with them, because it's so much more fun if we're able to do a lab, but that's how things are right now. Now, of course, you already know that a colony is a visible mass of cells that came from one cell. And of course, all those cells came from one cell. They're all genetically identical, so they're all clones. The cell that produces a colony is referred to as a colony forming unit, a CFU, and we'll see that term later. Now, a streak plate is a very important technique. It was so important that we would actually grade you on a streak plate. So in lab, you would have made a streak plate and you would have been graded on the quality of your streak plate, but you would have been given a second chance. Unfortunately, we're not able to do this, so we just have to talk about it. And we'll talk about this again in lab much later on. But once again, you have a sample that contains more than one microorganism. So a streak plate technique is a way to separate those mixed bacteria on the surface of the agar plate. And so the idea is you take one loopful of bacteria, and that's all you need is one loopful. You streak the upper portion of the plate. So these are all the bacteria that you're putting on the plate. You sterilize your loop, and then you drag it into that first zone, usually 2 or 3 times, and you're spreading out cells that were in zone 1, and you spread them out in zone 2. You sterilize your loop again, and you drag it into zone 2, usually more than one time, but the idea is you're grabbing a few cells from zone 2, and then you spread them out in zone 3. And so by the time you get to zone 3, you're actually depositing a single cell here, a single cell there, and they grow up to form discrete isolated colonies. And so in this way, you can separate more than one organism on the surface of an agar plate. Remember, each colony comes from one cell, so each colony represents a pure culture. And you can pick that colony off the plate and grow a culture of bacteria that contain just that one organism. So that's the benefit of the streak plate technique, is you can separate mixed bacteria on the surface and obtain discrete isolated colonies. So it's a very important technique. There is more than one way to do a streak plate. We're looking at the most common technique, right? The standard technique. But the idea is to separate the bacteria and obtain discrete isolated colonies. And that would have been fun for you guys to do. When it comes to preserving bacteria, we freeze them. And we freeze them really cold. right down to minus 95 degrees Celsius. So that basically stops all possible metabolic activity. We can freeze dry them, remove the liquid as we freeze them, so you end up with a dry powder. The benefit to freeze drying, the lyophilization, is that you can maintain those cultures at room temperature. Just add some sterile broth and they come back to life. At minus 95, you have to keep them at minus 95 until you're ready to warm them up and they come back to life. When it comes to reproduction, we're going to focus primarily on the bacteria, which divide into binary fission, but other microorganisms can bud, which is an unequal form of cell division. This group here, the actinomycetes, when you see the word mycete, you should always think fungi. The problem, though, is that the actinomycetes are not fungi. They were reclassified as prokaryotes because they don't have a nucleus. And all fungi are eukaryotic organisms. So actinomycetes were put into with all the prokaryotes. But the problem is they make reproductive spores like a fungus. They tend to be fuzzy and filamentous. And those filaments, when they break off, they can form new colonies like a fungus. And we'll talk more about those later on. But we're going to focus on binary fission in bacteria. So, when a bacterial cell gets ready to divide, it replicates its chromosome. It then forms a new cell wall in between the two chromosomes and the cells, and now you have two. So one cell becomes two. Two cells become four. Four become 8, then 16, 32, 64, 128, and so on and so forth. So what's happening with each division, with each generation? What's happening to the population?
They are growing exponentially.
Right, the population is doubling, and so that means the growth is exponential. So that means bacteria can grow very rapidly. They can go from one cell to a billion cells in 24 hours under ideal conditions. So they can grow very, very rapidly. Now, we typically grow bacteria on Petri plates, test tubes, flasks, and so we call that a batch culture. It's a batch culture because it's a closed system. Nothing is being added, nothing is being removed. So their entire world is that plate, tube, or flask. Under those conditions, bacteria go through 4 phases of growth: lag, log, stationary, and death. Don't confuse lag and log, right? And stationary students always forget stationary. So in the lag phase, the bacteria are not actively growing. What they're doing is they're sensing the environment. They're seeing what's out there, what foods can be used, what resources are available. They're turning on genes, they're synthesizing enzymes. They're getting ready to divide. Once they're ready, they start dividing, and growth is exponential. But nothing good lasts forever, right? Because they're in a closed system. They will start to run out of food, their environment becomes polluted and toxic, and the growth stabilizes. So they enter stationary phase, where the rate of growth equals the rate of death. So number of new cells being produced equals the number of old cells dying. So the overall number of cells remains constant for a short while. At this point, if there are bacteria capable of making spores, like Bacillus and Clostridium, they will start to make those spores because they sense they're going to die soon. So they will make spores. Eventually, though, the environment is just too polluted and toxic, or they run out of food and cells start to die, and death is actually exponential as well. So the bacteria die almost as quickly as they reproduced. Now, I do want to mention persisters. So a very interesting thing that happens, not just with bacteria, but scientists have also discovered this for types of cells, including eukaryotic cells. But a persister is a dormant cell, but it is not a spore. It's an actual cell. And the cell down-regulates its metabolism, slows everything down, and that allows it to tolerate an unfavorable environment, at least for a little while. And this presents a problem with antibiotic therapy, because normally, bacteria will actively try to resist the antibiotic, right, so they can survive. Here, the bacteria aren't doing anything to resist. They simply have slowed down their metabolism so much the antibiotic has no effect on them. So they're tolerating the antibiotic. And since we normally take antibiotics, for 5 days, 2 weeks, but a short period of time, those persisters can simply wait. And once you finish the antibiotic, they upregulate their metabolism and start to divide again. And so that's one of the contributing factors to bacteria being able to survive antibiotics, is because some of them just go dormant and wait till you finish the antibiotic, and then they start to reproduce again. We've also seen this kind of thing in cancer cells, in tumors, where not all the cells of the tumor are cancer cells. Some of them switch back to normal cells, so they're like a persister, which means the chemotherapy and the radiation doesn't work against them. And that's one of the reasons why some cancers come back, is because they had these persisters, these dormant cancer cells that kind of look like normal cells, so they were no longer killed by the chemo and radiation. So the concept of persistence probably evolved a very long time ago and is used by all kinds of cells, both prokaryotes and eukaryotes. So here is the growth curve for bacteria in a batch culture. Notice in the lag phase, they're not actively dividing yet. They're sensing the environment, turning on genes, making enzymes. Once they're ready, they grow exponentially. But nothing good lasts forever, right? They're running out of food, the environment's becoming polluted and toxic, so they enter into stationary phase where growth equals death. But at some point, their environment's just too toxic, they run out of food, start to die, and they die exponentially. So if it's your job to maintain cultures and you wait too long, you may lose your culture completely, right? Because they die out quickly.
So can you prevent dying in the stationary phase?
Well, this is what you can do. You can create a continuous culture. And so in a continuous culture, you remove part of the old culture and you add fresh nutrients. And that way you can maintain a constant state of exponential growth. And that's done with a device called a chemostat. And I ask students, but there's no time, what part of your body acts like a chemostat? And that's your gastrointestinal tract. Because you have a very large population of microorganisms there. You feed them, you add nutrients every day, and you remove part of the old culture every day, so you can maintain a constant population in your intestines. When it comes to measuring microbial growth, there are many different techniques. Some measure all cells, both live and dead. Others just measure live cells. And we'll actually do a lab on this one, so I'll talk more about that later. But we perform a serial dilution to dilute the original culture so that when we spread samples onto agar plates, we actually get individual cells that we can see. And so here would be the results. Way too many cells here. Here, right, they're spread out. We can count them. Here, we can count them, but there's actually so few, this plate would not be counted, because with all the experimental error involved, if there's too few cells, then it's just not accurate. So we look for plates which have between 30 and 300 colonies on the plate, and that's what we count. But we'll go into that later, and how to do a not too complicated calculation to figure out what the concentration was in the original tube by counting this plate, but we'll get into that later. So that's it for the lecture material. I do want to show you something, so I have to share my screen. It's on your handout. So let me see where the handout is. So at the very end of your handout will be a bonus question on the exam. And so the bonus question is calculating bacterial generation times. And here's some general formulas, but on my web page, there is a document on my web page calculating bacterial growth. So this goes through a more detailed example of how to calculate, and you have some problems, sample problems, with the answers, right, the solutions. So you should definitely take a look at that. It's something, if you want to do it, do it, but you don't have to because it's a bonus. So we'll definitely take a look at that sheet that's on my web page. Let's see. So here's my web page, lecture handouts, calculating bacterial growth. So, but any questions on the lecture?
Professor,
Uh-huh.
you're calculating bacterial growth. Are we turning it in or are we just doing it on our own?
Well, you learn how to do it, and there'll be a bonus question on the exam.
Okay.
And I believe the bonus is worth 5 points. And you get partial credit. All right. So I'm going to stop recording this right now so we can pick up with lab at about 1:05 in 10 minutes. Okay?